Good morning. I'm so pleased to follow Jan and also extend a very warm welcome to each of you for what I honestly can't believe is the 7th annual CPTR workshop, as Jan mentioned. I've been with you in this role for 5 of those 7 years and was an early participant on the pharmaceutical side in the formatory days. And as Jan says, this partnership over those 7 years has evolved as we really had hoped and dreamed that it would, more towards realizing a vision through some tangible, achievable goals that have been reached. So let me first figure out if my slides are up. There we go. There we go. So welcome again to the 7th annual workshop. I have always the privilege and the honor of kicking this meeting off by telling you about the fun stuff, all of the great work that you have accomplished over the past year. We intentionally gave ourselves a little bit of breathing room between our last workshop, which was in 2014, and today. And we did that because through our partnership with all of you, we had been working on some really important data sharing collaborations, as well as the evaluation of several drug development tool methodologies and building several modeling and simulation tools. We would really hoped that those programs would mature a bit over time so that by the time we came here with you, we would have some really exciting news to share about those programs, and I'm pleased to say that we do. So that was the spirit of why we took a little bit of a break from our regular fall schedule. We also wanted you to enjoy the cherry blossoms this year here in DC. So before I tell you about those specific areas of progress, I do want to start this talk with a thank you rather than bolting it on the very end like we typically do in PowerPoint presentations. First, a thank you, as Jan said, to all of you who have taken more than 4 days out of your life and work schedules to be here and be part of this collaboration. Were it not for you, all of the programs that I'll talk about today wouldn't be possible. So you give us so much of your time throughout the course of the year, but to be here for 4 days is a really huge investment. And thank you to all of you who are willing to be speakers and panelists. We've gotten so many wonderful questions from each of you over the last couple of months. I know how much preparation you've put into that, and so I'm really grateful. Last but not least, we have to thank the Bill and Melinda Gates Foundation Events Planning Team and our project management staff at Critical Path. Lindsay and our folks have done a tremendous job in making sure you all got here safely, you had a room to sleep in, and that we had an agenda to follow. So I'm really grateful for all the time that they've put into that. So I start my presentation with statistics that you all know very, very well, so I won't belabor them, but the idea that the global burden of tuberculosis in 2014, although we're making strides to bring those numbers down, is unacceptably high. And if we really hope to make a difference in that global burden, we need a new therapy and we need new rapid drug susceptibility tests in order to better treat these patients. So I start here to say that although we're 7 years in, the mission of CPTR is still incredibly relevant. One exciting fact of CPTR is that in the last 6 months, we have an incredible number of new members, many in the diagnostic space. A few of you here with us today, this is the first time you've really heard in depth about the CPTR initiative, and so I always like to start with a reflection on why we are here, and Jan touched on it a bit in his keynote. We just have some pretty animation here. So if our goal conceptually is to accelerate the development of that entirely novel drug regimen for TB, if we follow the current drug development paradigm and regulatory approval pathway, right, this is an unacceptably long period of time between now and the realization of that new regimen, combining things only at the very end of all of their successful individual development cycles. The question in those early days, as Jan said, as we had the big vision thinking, is how do we get here, and what is the role of this collaboration in the accelerating that pathway? So our goal is to do something more like this, right? Through early combination testing strategies to move these drugs in parallel as part of an entirely new regimen. And again, this is a really big set of challenges we face. So where do we start with the work of CPTR? As Jan mentioned, we have invested a lot of our early prioritized work and resource around understanding first the methodologies that exist in the drug development space for TB. We have some great tools, but now with some new data, could we learn a little bit more about those methodologies and make them better? And where we don't have the tools that we need to help improve regimen development, can we create them? And we've been in a really great position over the last couple of years to do that good work because so many of you have generated contemporary TB clinical trial data in the last 5 or 10 years. And we have equal amounts of robust preclinical data that we can use as well. And I'll talk to you about how that data sharing and data collaboration has really moved our initiative forward, really in the last 18 months since we spoke to you last. So our goal and a big area of emphasis has been on this critical roadmap toolkit. So what are the best tools to make drug development decisions in the TB regimen space? We're always up against the fact that we have a very fragile drug development pipeline for TB. Many companies have exited, and so we are going to spend a lot of time in day 2 talking about what that drug development pipeline looks like, and equally as important, spending time to learn a little bit about what the discovery pipeline looks like in TB, because that's really what's going to refuel the work of CPTR. And as Jan mentioned, we realized 3 years ago that there was a gap in our thinking if we did not address the issue of rapid drug susceptibility test development, that if we are successful in getting new drugs and regimens forward, if you can't deploy them on first dose to the patients that need them most, we have a gap in our strategy. So hence, starting that component of the consortium, you'll hear a lot about that work in days 3 and 4 of this 4-day meeting. So what I want to do here is just pause to say a lot of what you're going to hear me talk about in terms of methodologies and data platforms, this is work that's ongoing within our Regulatory Sciences Consortium, but there's a lot of other great work, very important work that's ongoing in the other 3 arms of our large initiative. So you'll hear a little bit about later today some work in our research resources arms from our Global Regulatory Pathways Group and our stakeholder and community engagement team. I mentioned to you the Drug Development Roundtable that's led by the TB Alliance. You'll hear about that tomorrow. And then in days 3 and 4, our Rapid DST Consortium and a big collaboration around a new data platform there. So to focus a little bit on these tools that we talk about. So I've oriented this slide, hopefully purposefully, and hopefully you can read it. The bottom 3 activities on this slide are all data collaboration initiatives that are either launched directly through CPTR or have come about as part of a partnership with collaborators within this public-private partnership. So our clinical trial data platform, you've heard me speak about that many times over the past 5 years. First data contribution to that platform was the bedaquiline data set. This is the data platform that's really sort of proprietary and only accessed by specific working groups within this initiative. That's really driven a lot of the work that we've done around data standards, biomarker evaluation, et cetera. We have a large data platform that you'll hear about in day 3 and 4 that's whole genome sequence level data, partnered with phenotypic data and patient outcome data where it's possible, really driving the assay development work within the Rapid DST Consortium. And an exciting new collaboration between the Critical Path Institute and the WHO TDR, which has brought together the 3 quinolone-containing phase 3 clinical trials in an aggregated form to be publicly accessible by everyone. So I'll talk to you a little more about that in a coming slide, but to let you know that a lot of that progress has happened in the last 18 months since we've spoken. I put these components on the bottom of the slide intentionally because it's these data platform initiatives. Okay, these aren't databases where we just store data. We've put them in a place where they can be aggregated and made useful, and that's driving the work we're doing in modeling and simulation. You're going to hear a lot about those quantitative pharmacology tools later on today. We also access these data to help us do a robust evidence-based methods evaluation for a lot of the preclinical model work that we've done within the initiative. We can build data platforms. We can evaluate preclinical methods. We can develop new modeling and simulation tools. But if they're not useful to the stakeholders in this room, if you don't understand how they can be useful as part of your programs, if we don't teach and train you how to use them, we have not done our job. And so part of the reason that we meet today, before these tools are 100% complete, is to ask your thinking and start a conversation with each of you about how we can do a good job of deploying and implementing these methodologies. And so I hope that will be part of the discussion that we have in the coming days. just a graphical representation about the use of these aggregated data platforms. I know sometimes it's hard to see the relationship of the organizational structure of CPTR and the actual data work that we do. So this is just to show that we have those 4 large data platforms that I mentioned. What I didn't mention in that last slide is that we do have a large preclinical data repository that began largely with the work that we're doing in the in vitro hollow fiber system. Now we are populating that data platform with work that we're doing on the sterilizing mouse model, in large part with Eric Nurenberger and Ann Lennertz. And that all of these data provide us the supportive evidence base to do methods evaluation, development of new modeling and simulation tools as part of our research resources group, and that the Rapid DSD Consortium is largely leveraging the relational sequencing or ReSeq TB data platform that I mentioned. All of this is made possible through the use of the data standard, the TB data standard that was developed through this CPTR partnership several years ago, led by the team at Critical Path and CDISC. Bess Leroy is here in the room and has done a tremendous job with that data standard. I know that we always say data standards aren't sexy and interesting, but I'm going to tell you that this tool that was developed years ago can be one of the most useful tools to this TB community, because it helps us to make better use, a bigger return on investment for all the important clinical trials that you run. We have, in the past year, updated this data standard to include rapid drug susceptibility test terminology as well as pediatric terminology. So we hope that will increase its value and use in the TB community. So the idea about the data standard is that it allows you to easily aggregate or pool data across lots of different institutions, lots of different clinical trials, so that you can see subtle signals you wouldn't see in a single clinical trial. So again, this is just my pitch that it's there and usable. We have all of the information you need to use it posted on the CDISC website, on our own website for cptrinitiative.org, and we would be very happy to teach and train as needed on that data standard. So just a couple of slides, and I promise I will be brief, on these data sharing initiatives. So I'll begin with the TB Pacts, the platform for the aggregation of clinical trials for TB. This is the partnership between Critical Path Institute and the WHO TDR to make available to the entire community the 3 quinolone-containing Phase III studies, Remox with Rifaquin and Oflotub. On April 15th, we will announce the launch of this data platform. We will be sending out a press release, and all of you in the room will be getting information about how and where to access these data. The process for access is very simple. I mean, the basics are you have to be part of a qualified research institution, a legitimate institution. You have to provide us with a summary of the type of work you're seeking to do. We would like to be able to post that on the website so that researchers in the community can know if some of the ideas they have are already being investigated, if they might want to go another direction. But the idea is that this is a publicly accessible database made useful and increasing the return on investment that was made in generating all of the data from those 3 clinical trials. So that will be coming forth April 15th. The ReSeq TB Global Partnership. So this is that whole genome sequencing driven data platform that we talked about at our last meeting in 2014. It was one of those aspirational goals at the time, and there was lots of conversation and discussion and dialogue at that meeting about how we could be successful with an initiative like this, where we might face challenges, and I'm very happy to tell you this platform is alive and well. It's fully architected. We have 3,500 isolates worth of information. That data platform has been accessible to the CPTR consortium for the better part of the last year. We've gotten a lot of good feedback on how to optimize that website, and you'll hear a lot from this team and the expert committee that drives it in the coming days of our initiative. So data for data's sake is not interesting. Nobody needs another data repository. But I want to really emphasize the value of the data sharing collaborations that we've driven through CPTR and our growing partnerships. It's how we are leveraging data to create tools that didn't exist before. And a big part of the way that we are doing that within CPTR is within our modeling and simulation program. Again, you're going to hear a lot from our panelists today on these now 7 programs that are being driven through CPTR. We are leveraging these data to develop everything from optimized in vitro methodologies like the hollow fiber system that we've talked about at great length in previous meetings, to validate other preclinical methodologies like the sterilizing mouse model. We've developed a PBPK model so that we can understand drug distribution of TB lungs in an adult patient infected with tuberculosis. A systems pharmacology model where we're working to understand the mechanism of action of these new drugs and how that might affect the way we would design a clinical trial, all the way out to a penultimate clinical trial simulation tool. These are critical tools, we think, in driving the way we make decisions about what that next new regimen should look like. And all of that is coming through the collaboration of data that you all have contributed and partnered with us on. So I've talked you through most of the bullets on this slide. Just wanted to leave you with some information on where we've come and where we expect to be going in the coming year. I mentioned that we've updated the TB Data Standard version 2.0 is ready to go, including the terminology that I had mentioned to you. It's important to note that for regulatory-driven programs, the FDA is going to require the use of CDISC Data Standards by 2017 for all trials for which they are evaluating. So we would like to see the same implementation and rigor with publicly funded trials going forward as well. We've talked in previous years about the qualification of the hollow fiber system model with the European Medicines Agency. What I will tell you is we've done a tremendous amount of work since the last time we met with the Baylor laboratories on the hollow fiber system, further validating it and understanding where there are sources of variability in these very complex studies and how to mitigate that variability. We think it's an incredibly powerful tool as part of a larger toolkit in the preclinical TB drug development space, and we are funding a large second phase of work, which you'll hear about more later today, to do some prospective investigation with new drugs of interest and new drug combinations. On the rapid DST side of things, there was a partnership between CPTR, the World Health Organization, and the CDC to come to some level of agreement about standardized terminology for surveillance studies. That work, that meeting was very successful, and they're progressing towards recommendations in the coming year. All of the details behind what I've just mentioned have been published in the last 18 months. We have 18 publications in 5 major supplements across major journals, spanning everything from our hollow fiber and regulatory work to some of the modeling and simulation work to some great editorials that Marco and others have written in the DST space. So where do we plan to be by the next time I'm up here again? So these are the things you should be holding me to, as well as yourselves. The relational sequencing data platform is just making huge strides, and by this time, at the end of 2016, actually, we hope to be up to 10,000 isolates worth of information, and we want to continue expanding this. So with all our data programs, if you learn about these over the coming 4 days and want to contribute data or know a great data contributor that we haven't reached out to, we would love to hear from you. I've mentioned the TB Pacts platform, April 15th. That will be live and well. We will have 5 of our 7 modeling and simulation programs complete by the end of 2016. So you should be hearing from us about teaching and training opportunities on those methodologies. And the newest of our modeling efforts will be a pooled analysis that will be driven by Rada Savic's lab on those quinolone-containing data sets. This is a collaboration between the World Health Organization and the CPTR initiative. We are very excited to get that launched and started, largely driven by needing to access this data platform soon. So we are working on that. So finally, I can just breathe a little bit, tell you about our program, and give you a few housekeeping details before I introduce our keynote speaker. So everybody is asking about Wi-Fi access. Yes, we have it. They ask that you first try, if you're staying in the hotel, to use your hotel information to log in. If for any reason you cannot gain Wi-Fi access that way, both the username and the password for Wi-Fi access in this room is CPTR, all caps, 2016. But I will make a plea as you all rapidly start typing that into your laptops. We really do encourage your participation in this workshop. We call it a workshop for a reason. We don't want this to be a series of presentations. We really want dialogue. So please help me out. Also, silence your phones. This is a big room, and so if a bunch of phones start going off, it gets a little bit distracting for the panelists and presenters up here. You will see when you came in today, you were provided a pretty extensive program. Our program has gotten so large, we have to give you a snapshot of our program, which is in the front page of the booklet. So that'll give you a sense of where to go and where to be over the course of the next 4 days. We are, over the course of these 4 days, videotaping all of the presentations and the dialogue that will be ongoing in the breakout sessions. I just want people to be aware that videographers will be around. We will be making available all the presentations you see over the course of these 4 days on our CPTR website as quickly as we can following the workshop, and we'll notify everybody when those are there and available. Lastly, you were provided a USB drive that is a lovely bracelet when you entered. That has loaded on it a digital copy of the program, but also some important fact sheets that we have about the data platform work that we've just talked about and the CPTR initiative in general. So we hope that you'll find that helpful following the workshop. So with that, it is my absolute pleasure to introduce to you our keynote speaker today. Dr. Mark Sulkowski is professor of medicine at Johns Hopkins University and the medical director of the Viral Hepatitis Center in the divisions of infectious diseases, gastroenterology, and hepatology. His research there focuses on the development and delivery of novel therapies for treatment of hepatitis C infection for people with and without HIV co-infection. He is a clinical pharmacologist, and his work was integral in the development of the most recent hepatitis C regimen. So quantitative pharmacology best practices, understanding viral kinetics, understanding how the mechanism of action of new drugs is important in treating infection and how different drugs may interact with one another was critical for designing that therapy. As I mentioned, his work was integral into that story, and we just felt, given the timing of where CPTR is at, the work that we are doing on our quantitative modeling and simulation programs, it was incredibly timely to hear about the hepatitis C story. So we've asked him to tell us a little bit about that story and also to challenge each of us in the room to think maybe a little bit differently about how we've approached TB regimen development in the past. So please join me in welcoming Mark Sulkowski.